This video is part 2 of a 2-part introduction to photosynthesis for AP Biology. In the previous video, we looked at the overall equation for photosynthesis. Carbon dioxide and water enter plant, and in the presence of light, that plant is able to synthesize the sugar, glucose, and releases oxygen as a waste product. We also looked at the anatomy of a plant, specifically the anatomy of a chloroplast. We're now familiar with a thylakoid, a grana, the stroma jelly that surrounds the chloroplast, and that this chloroplast is where sunlight, carbon dioxide enter, and energy and sugar exit. Now we need to answer the how question. How are plants able, through photosynthesis, to synthesize sugars that they can then break down during cellular respiration to generate ATP? Photosynthesis is made up of 2 overall reactions. The first one is referred to as the light reactions, or to be more specific, the light-dependent reactions. Why? Because these reactions require light to occur. This is where plants are going to be able to convert solar energy into chemical energy. Specifically, to synthesize 2 molecules, or regenerate them to be more accurate. This is where we're going to be able to generate ATP and an electron carrier called NADPH. NADPH works the same way as NADH in cellular respiration. It's a molecule that's able to capture electrons and hydrogens and transfer them from one location to another. The second reaction that happens in photosynthesis is the Calvin cycle, also known as the light-independent reactions. These reactions can occur with or without sunlight. Sunlight is not relevant. It is this reaction where we're going to build sugars. This is where we're going to be able to take carbon from CO2, use that energy that we developed in ATP and NADPH from the light reactions, and synthesize glucose. I want to first give you a million miles away view of just what's going in and out of each reaction. So let's start first with the light-dependent reaction. The light-dependent reaction happens inside the thylakoid, inside the chloroplast, and that's what you see in this diagram. The big green bean is a chloroplast, the small green circles are thylakoids. This reaction starts when water and light enter the thylakoid. There, a series of reactions will occur. I'll talk about them later. Right now, I'm just symbolizing it with a rotating explosion. And out comes oxygen as a waste product, and ATP and NADPH, molecules that can be used for energy later. After this, we can now do the light-independent reactions. In the light-independent reactions, the energy in ATP and NADPH are going to be used to take carbon out of CO2. Through reactions we'll detail later, out comes a synthesized glucose molecule, and the now oxidized ADP and NADP plus return to the light-dependent reaction, where they can be regenerated again. We can add more phosphate and more hydrogens to regenerate into ATP and NADPH. I'm going to start first by looking at the light-dependent reactions. As we go through the steps, please keep in mind the overall goal. The goal of the light-dependent reaction is to convert NADP plus NADP into NADPH and ATP. Oxygen is just released as a waste product as a component of this process. I'm going to detail all the steps involved, but first I want us to watch an animation of it occurring.
Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars.
A reminder that a photosystem is just a collection of pigments such as chlorophylls A and B and carotenoids that are able to capture energy from light.
The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first.
The reason that is the photosystems are named after their in order that they've been discovered. Photosystem 1 was discovered first, photosystem 2 was discovered second, but it turns out that photosystem 2 is the primary driver, the initiation of this process.
The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecule,
we then transition to the light-independent reaction. But first, I want to review everything that you saw with the light-dependent reaction. So let's go step by step. The first reaction you saw was that of photosystem 2. So in photosystem 2, a series of reaction occurs. First, water is split by an enzyme. In the video, they mentioned that it's oxidized. When water is split, that causes the electrons from that splitting to move up into an electron transport chain. It also releases H+ ions, which will build up inside the thylakoid membrane, creating a concentration gradient, meaning we have more hydrogens inside the thylakoid than we do outside. Those electrons get excited by light. This is photosystem 2, so we're dealing with P680. And when they get excited, that energy enables them to move down the chain and enables them to power the pumping of hydrogen ions from outside the thylakoid into the thylakoid space. Again, adding to that concentration gradient. Why all this effort to build a concentration gradient? Well, in our next step, we're able to use that concentration gradient to power the enzyme ATP synthase. Since we have a proton gradient now inside the thylakoid, it'll diffuse out, and when it does that, it turns the turbine of ATP synthase and generates ATP. That ATP then goes out into the stroma where it can later be used in the Calvin cycle. With the last step, those electrons that were moving down the electron transport chain, they didn't go away, they're still there, but they're no longer energized. They've spent the energy they got from light. So when they reach photosystem 1, they again get hit by light, and that energizes the electrons and excites them. This happens with pigments that are absorbing the frequency 700, so we often refer to photosystem 1 as P700. When those electrons are excited, they can be used by an NADP+ to attach two hydrogen ions to form two NADPH, our electron carrier, which will then go off and be used in the Calvin cycle. A quick comparison of electron transport chains between photosynthesis and cellular respiration, because I think it's helpful to see how these processes are very similar, they're just reversed. What you saw with the light-dependent reaction is that water was split. And a reminder, in cellular respiration, oxygen is the final electron acceptor and it synthesizes water, so water is produced. With the light-dependent reaction, hydrogen ions diffused out of the thylakoid. We built our concentration gradient in the thylakoid and it diffused out into the stroma. With cellular respiration, we had the opposite direction. We had H+ ions diffusing into the mitochondrial matrix, into the innermost part of the cell structure. With the electron carrier in The light-dependent reaction, we produced it. We made NADH. But at the electron transport chain in cellular respiration, we spent our electron carriers. We broke them apart to get the hydrogen ions used for ATP synthesis. Notice the mirroring here. They're the exact same processes. They're just reversed. And that's an end product of evolution. So I want us to remember what it means to be a plant. You're going to need to be able to produce all of your own organic molecules, carbs, lipids, proteins, nucleic acids. You don't eat so you can't get it from food. You also need to be able to store chemical energy produced from the light reactions in a way that's stable and can be moved around. So with the light reactions, what you saw was we got energy in the form of ATP, and we got some reducing power from NADPH. We got some money in the bank. So now we can use this energy that we've created to build stuff. We've now taken care of the photo and photosynthesis with the light-dependent reactions. Let's now get to the synthesis, the building of sugars. This is super helpful because we have raw materials to draw from if we have an energy source. Plants have a source of carbon in the atmosphere. There's plenty of carbon in CO2. They just need the energy to extract it out. So with our next step, the big overarching purpose is to do something called carbon fixation, meaning we're taking carbon from CO2 in the atmosphere, fixing it or gluing it together to make a larger energy molecule such as glucose. NADPH is going to provide the reducing power for that to happen. And what's great about NADPH and other electron carrying molecules is that they can be recycled and reused, so it's not a big expense for an organism like a plant. So now we've left the light reaction, which occurred in the thylakoid. Everything from here on out with the Calvin cycle is occurring in the stroma. We're going to take the same approach this time. I'm going to show you the overarching purpose. We're going to watch an animation and then we'll go through the details. So for the light independent reaction, also known as the Calvin cycle, the entire point of this is to use that energy that we got from the light dependent reaction in ATP and NADPH to remove carbon from CO2 in the atmosphere to synthesize glucose. So let's see it animated and then we'll go through it in greater detail.
Are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate.
A note about glyceraldehyde 3-phosphate, G3P, you've seen that before. You've seen that in glycolysis when we first split glucose. With G3P, if you combine two G3Ps together, you get one glucose. This process is rather confusing, but keep in mind for every G3P it makes, if it makes two of those G3Ps, the plant has now synthesized one glucose.
The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate.
Notice that what happened there is with 3 turns of the Calvin cycle, 1 G3P was pulled out of the reaction. 2 G3Ps are needed to make 1 glucose. If I get 1 carbon from each CO2, and I need 3 carbons to make 1 G3P, that means I need to do the Calvin cycle 6 times to get 6 carbons for 1 glucose.
The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose.
So that's the Calvin cycle. It's a lot to take in. Let's review it again. So here are my Calvin cycle, and again, this is happening in the stroma of the chloroplast. It's a cycle, so we're beginning with the end product, and our end product is RUBP. RUBP is a 5-carbon molecule. The plant's going to take in 1 molecule of CO2, has 1 carbon. An enzyme called Rubisco is going to be the enzyme that makes this happen. Ribulose bisphosphate carboxylase, Rubisco in human, is going to be able to grab a CO2 molecule, grab an RUBP, and bind them together to make a 6-carbon molecule. Now, this diagram I'm showing you is representing 3 turns of the Calvin cycle at once. Here you can see 3 carbons that are entering from CO2 and 3 RUBPs. That's the step that we call carbon fixation. We have fixed a carbon from the atmosphere into the chemistry of the plant. Now that we've done that, we need to reduce. That 6 carbon is going to be broken down into PGA, a bunch of 3 carbon molecules, and we're going to use ATP to do that. A reminder that when you add phosphates to a molecule, it destabilizes it, just like it did in glycolysis. By breaking it apart and using that NADPH energy, we're reducing the carbon molecule, and we end up producing a series of G3Ps. With 3 spins, we make 6 G3Ps. One of those G3Ps is going to be removed out of the cycle and used to make glucose. The remaining 5 remain in the cycle because they're needed to regenerate the RUBPs that we started with. That's why the last step is called regeneration of RUBP. So to review this again, this is showing 3 turns of the Calvin cycle in the stroma. 3 carbons enter and are grabbed by an enzyme called Rubisco. Rubisco grabs the carbons and the RUBP and fixes the carbons or adds them into the cycle. We call that phase carbon fixation. After we fixed our carbon, energy from ATP and NADPH are used to reduce the molecule. We're removing components. We're adding different components. Reminder that the phosphates are there to destabilize the molecule, and we use that to generate 6 G3P. One of those G3Ps are removed. The remaining 5 are used to regenerate RUBP in the last step, so we can repeat the cycle again. Now, this was only 3 spins, so I made 1 G3P. I need to do a total of 6 spins to generate 1 glucose. So this happens 6 times simultaneously for 1 glucose. Hopefully you remember G3P from glycolysis. That was that molecule that was split. So why we want to form glycolysis, G3Ps can also not be glued together to make a glucose. They could be directly put into glycolysis or used to build other compounds. A little bit about Rubisco. Rubisco is one of the most abundant enzymes in a plant. Why? Because it's essential for photosynthesis, and without photosynthesis, plants can't get the sugars they need in order to do cellular respiration and get the energy required to survive. Here you can just see an accurate rendering of what Rubisco looks like. So to do some accounting, 3 turns of the Calvin cycle makes 1 G3P. So 3 CO2s in, 1 G3P out. We need 6 turns to make 1 glucose because we need 6 carbons for glucose. Glucose is C6H12O6. Throughout this whole process, we're actually using 18 ATP and 12 NADPH to make 1 glucose. That's a lot of activity for the light-dependent reaction to generate that much. So in summary, the light reactions produced ATP and NADPH, and consumed water to build a concentration gradient. To make that ATP and NADPH, oxygen came out as a waste product. The Calvin cycle, we consumed CO2, we fixed it, generated G3Ps. We had to use our ATP, so now we're left with ADP, and we had to use our NADPH, so we're left with NADP. This is again just showing you a summary of the light reactions. We had water and sunlight in, NADPH and ATP came out. Oxygen was lost as a waste product. That's because of the work of photosystem 2, splitting water and moving electrons, creating the gradient. ATP synthase being used to generate ATP, and photosystem 1 regenerating NADP plus 2 NADPH. Calvin cycle, we used that energy, CO2 went in, and we built sugars. So to connect the two, because remember, plants do photosynthesis and cellular respiration. With photosynthesis, we're generating sugar and oxygen. Those are the reactants of cellular respiration. So now the plant can use both of those to undergo cellular respiration and generate ATP, and the plant will release CO2 and water as a waste product, which are the reactants of photosynthesis. So it is this biochemistry, this dynamic between photosynthesis and cellular respiration, that explains the evolutionary and ecological relationships we'll explore later in this class as to why the animal kingdom is so inextricably dependent on the plant kingdom and vice versa. So hopefully by the end of this series of videos, you're able to answer questions like this. In the case of photosynthesis, where did the CO2 come from? Where did it go? Where did the water come from? Where did it go? Where did the energy come from? And what was it used for? What is that sugar, that glucose, going to be used for? And where did the oxygen come from? Where did it go? And everything else not involved. Hopefully now you have an understanding to explain broadly how photosynthesis works and specifically how these atoms of carbon dioxide, water are being converted into a sugar and oxygen molecule. I hope this was somewhat helpful in your understanding of photosynthesis, and I'll see you next time.